This video is going to be all about the Krebs cycle, and to start off with, I just want to make sure that you know all the names you may see the Krebs cycle described as. So you may see it described as the tricarboxylic acid cycle, the TCA cycle, or the citric acid cycle. Now, a classic type of question that you'll see is, where does blank occur? Like, where does glycolysis occur, or where does the Krebs cycle occur? And the Krebs cycle occurs in the mitochondria. And this slide is really just meant to be an overview to give you an idea of where the Krebs cycle fits into the big picture of metabolism. So this is a fatty acid here. This is a 16 carbon fatty acid, and then we do beta oxidation on it, and then we get acetyl-CoA, a bunch of acetyl-CoA. So we'll get 8 2-carbon acetyl-CoAs. And then acetyl-CoA jumps into the Krebs cycle. And then the Krebs cycle is going to make some energy intermediates called NADH and FADH2, and it's also going to make some ATP. And then these high energy electron carriers, NADH and FADH2, are going to donate their electrons into the electron transport chain. And then one kind of tricky thing to keep track of is that the final step of fat metabolism is done by the Krebs cycle. And so when you cut apart the fat through beta oxidation, you're left with 2-carbon acetyl-CoA. And then through the process of the Krebs cycle, that acetyl-CoA goes away. And so after we're done with the Krebs cycle, what was remaining of the fat is now gone. And all we have are these energy intermediates. So I'm organizing the rest of the lecture into the different question types that you typically see from the Krebs cycle. So we're going to see counting carbon questions. We're going to see questions asking you about intermediate names, questions about enzyme names, questions about inhibitors of the Krebs cycle and regulation of the Krebs cycle, and then the energy yields or the energy math, and then some etc. questions. So with the Krebs cycle, we're starting with a 4-carbon intermediate and joining it with a 2-carbon intermediate. The 4-carbon intermediate is called oxaloacetate, and then the 2-carbon is acetyl-CoA. And we join those to make citrate, which is in part, where that name, the citric acid cycle, comes from. And so we have a condensation reaction of oxaloacetate and acetyl-CoA, and I'll abbreviate oxaloacetate as OOA. So now we're going to talk about the intermediates, and I found this really great memory aid to keep track of the order of the intermediates and their names. So it goes, citrate is Krebs starting substrate for mitochondrial oxidation. Now let's talk about some of the important intermediates. The first one we'll talk about is oxaloacetate. So you got to know that oxaloacetate plus acetyl-CoA is the first step of the Krebs cycle. And then you have to know the connection between oxaloacetate and aspartic acid. So I have this arrow here showing that the two are interconvertible. So you can take aspartic acid and convert to oxaloacetate and also do the reverse reaction. So aspartic acid is the most immediate source of oxaloacetic acid during the Krebs cycle. And I got to throw a memory aid in here because, otherwise, how in the heck are you going to remember that? So we're going to think, ooh, nice ass. So OO for ooh and A for ass, aspartic acid. For alpha-ketoglutarate, you have to know the enzyme that makes this one. Isocitrate dehydrogenase produces alpha-ketoglutarate. And then you got to know which intermediates are acids. And so oxaloacetic acid, also known as oxaloacetate, and alpha-ketogluteric acid, also known as alpha-ketoglutarate. So let's just talk about how this slide is organized real quick. On the left here, numbered 1 through 8, I have the intermediates from that little memory aid that we talked about. And then I have the enzyme that makes that intermediate. So for example, here, citrate synthase makes isocitrate. And then out of the reaction comes CO2 and NADH. Now, we're going to look at this one, isocitrate dehydrogenase. This is an important one to know. You got to know that this one makes alpha-ketoglutarate. So you may see a question that asks something like, isocitrate dehydrogenase makes which intermediate? And you'd have to know that it makes alpha-ketoglutarate. And then here's an important point, that this is the final step of